Good morning, class. Today's topic is recombinant DNA technology. In this topic, we have to study about the tools and recombinant DNA technology. What are the tools? The tools used in this technology is the recombinant DNA technology is accomplished with the tools. First tool is known as the restriction enzymes. Restriction endonuclease and restriction exonucleases. There are two types of enzyme responsible to cut down the nucleus, to cut down the chromosomes at the desirable point. So, restriction endonuclease enzyme is responsible for the recombinant DNA technology, first tool. Second tool is the polymerase enzyme responsible for the polymerase reactions. Third one is the ligase enzyme responsible to join the cutting pieces of the desirable DNA. The vector is used as a vehicles for joining of this desired piece and the host cell for the replication for making so many copies of the desirable genes or the genetic products. So, number one is the restriction enzyme. What are restriction enzymes? Restriction enzymes which is used to make cut in the DNA in the recombinant DNA technology. So these are the group of the enzymes which is used to cut the DNA in a small pieces for as per requirement. In 1963, the two enzymes were isolated in form of this restriction enzymes from the E. coli bacteria, which is responsible to restrict the growth of the bacteriophages. The bacteria-eating viruses, which is restrict growth of the this virus on the E. coli bacteria by the presence of the two types of enzymes, which is isolated in 1963. One enzyme that is added methyl group to the DNA and other cut DNA, and that was named the restriction endonucleases. So, two enzymes isolated from this bacterial cell. The main function of the restriction endonucleases depend on a specific DNA nucleotide sequence. Actually, the main function of the restriction endonucleases, each and every endonucleases enzymes is depend upon the specific DNA nucleotide sequence, means the function on the very specific DNA nucleotide. First endonucleases isolated was the HIND2, HIND2. It is studies proven that the HIND2 always cut the DNA at a particular point by recognizing a specific sequence of near about a 6 base pair. The 6th base pair is responsible, and this enzyme have to cut down by studying the recognition sequence of this enzyme. This restriction enzymes belongs to the nucleases. Actually, the group of this enzyme is nucleases, means the class of the enzyme it's two types. One is the restriction exonucleases and another is the restriction endonucleases. The exonucleases remove the nucleotides from the end of the DNA. Actually, if we have to remove the end part of the DNA, end part of the nucleotides, then the restriction exonucleases is responsible. But if we have to cut a specific position within the DNA in between the nucleotides, we have to use the restriction endonucleases. While naming the restriction enzymes, the first letter represent the genomic name and the second two letters represent the specific name of the prokaryotic cell from which the DNA was isolated. Means the DNA or the enzymes was isolated from the prokaryotic cell. The two letters, first letter denotes the genomic name and second two letters represent the specific name. How restriction endonucleases act? How it is going to act? The restriction enzymes in respect DNA sequence to find its specific recognition site. This DNA sequence to find the very specific recognition site and to bind and cut the specifically each of the two strands of the double helix at their sugar phosphate backbone bond. The restriction enzymes recognizes the specific palindromic nucleotide sequence. Palindromic means if we are going to start to read from left to right or right to left, the sequences are the same. It is known as the palindromic nucleotide sequences. Since we know that the DNA is a double helix structure, so palindrome in the DNA sequence of the base pair that react the same on the two strand when orientation of the reading is kept the fifth to third end in the both strand. Means if we have to study, we have to notice the nucleotides from fifth to third end direction. The same is there, and if we are going to read the fifth to third end in the double helix structure, means anti-parallel of these two, the same structure is there. For example, you can see here the GAA-TTC from fifth to third end, and if we are going to read this complementary base pair CTT-AAG, and if we are going to read these base pair from the again fifth to third direction in the second helix structure, it is the same GAA-TTC. It is known as the palindromic sequence. So if the restriction enzymes cut the strand away from the center of the palindrome sites between the same two bases of the opposite strand, leading to formation of a single strand portion, this end is known as the sticky end. So sticky end hydrogen bond with their complementary cut the counterpart DNA ligase. Here you can see in this picture DNA duplex, and after the function of the restriction enzymes, you can find here the two strands having the two sticky ends is there, and it have to join each other with the help of the DNA ligase enzyme. Some restriction enzymes are here. Number one is enzyme, the means name is the ECORI, ECORI, and the source organism is the means the extracting organism is the Escherichia coli, E. coli, and the restriction recognition site in the double standard, you can say find out the GAA-TTC, and here if we are going to cut it, the structure of the cleaved portions, where cleaved means from where this enzyme is going to act, the cleaved area is overhang, means G, and fifth and AA-TTC you can find over here, and in the PSTI it is then another enzyme, and it's extracted from the Providencia stouti, and this you can find here 3 base pairs is along there. This is the C overhanging. So this is the structure of the cleaved protected areas double in the after cutting of this double helix stranded, means the point from where it is going to be at. Third one is known as the SMAI, that is the Serratia marcescens, and the third one is the HAELLI, that is the Haemophilensis agitapis. These are the blunt end, overhang end, and the blunt end. The cutting portions in the black dot, this black dot is for the from the portion where this is going to be cut out, means the restriction recognition site of the double helixes DNA in all these enzymes. Now, what are the application of this restriction endonucleases? This main application is in the formation of the recombinant DNA. Used of the field of the genetic engineering to form the recombinants or recombinants, the molecules of the DNA, composition of DNA from the different sources. Same restriction enzymes from the vector and the source DNA. The same kind of sticky ends we have to find it, which can be joined with the help of the DNA ligase. This picture is taken from your NCERT book. This is the action of restriction enzyme EcoRI. The enzymes that cut both the DNA stand at the same site, vector DNA and the foreign DNA, GAA-TTC. And after cutting with the help of the EcoRI, you can find the sticky ends. With the help of the DNA ligase, it is joined here and form the piece of a DNA that is known as the recombinant DNA. This diagrammatic representation of the recombinant DNA technology, it is a diagrammatic representation. Suppose that here is the foreign DNA, have to cut it into small pieces with the help of the molecular scissor or the endonuclease enzyme. And the vector DNA, that vector DNA is taken from the plasmid of any bacterial cell, the extracircular DNA found in the cytoplasm of the bacterial cell, and have to cut it a portions in which the same restriction enzyme cutting both the foreign DNA and the vector DNA at a specific point, and with the help of the ligases enzyme join the foreign DNA to the plasmids, and now this plasmid is have to introduce into a bacterial cell by the process of the transformation in the E. coli bacteria. When E. coli bacteria divide, it have to make so many copies of this foreign DNA which is going to be ligazed on this plasmid DNA. So this is the diagrammatic representation of recombinant DNA technology. Thank you class, and have a nice day. In the next part we have to study the different tools used in the recombinant DNA technology. Thank you and have a nice day.